Okay, hello to everyone and welcome to this Early Career Academy webinar today. So it is our great pleasure to welcome Professor Mikael Rydén and docent Niklas Mejhert from Lipid Laboratorium at the Department of Medicine from Karolinska. So they are world's leading experts in human adipose tissue biology. And Professor Rydén was also recently awarded by the prestigious Camillo Golgi Lecture Prize at the EASD annual conference. So their research bridges molecular, cellular, and clinical studies to understand how adipose tissue contributes to metabolic diseases. And this has been instrumental in defining adipose tissue as an endocrine metabolic dynamic organ. And today the presentation is about high-resolution approaches to study white adipose tissue in health and cardiometabolic diseases. And in here they describe how high-resolution spatial approaches applied to human white adipose tissue from clinical studies can be used to uncover the cellular organization and how by integrating spatial transcriptomics and complementary profiling techniques, we can better understand adipose tissue biology in health and disease. So please, the floor is yours, Mikael and Niklas, and thank you for coming to present to us.
All right. Thank you, Sydney. Thank you, Alfredo, for inviting us. So again, this is a joint effort that we're going to subdivide into different parts. I'm going to start and end the presentation, and then Niklas will take the middle part, the juicy part. So I think it's important to, since we're not sitting in the same room, we will have to stop and switch presentations at the in the middle of this. I hope you're fine with that. I should also emphasize that we are also affiliated to the Steno Diabetes Center in Copenhagen, both me and Niklas. So these are my declarations of interest as a healthcare professional. I don't own any stocks in any company, any drug companies, so nothing is related to this presentation. All right, so the outline of what we're going to cover is first, I'm going to give you a very, very brief introduction of white adipose tissue dysfunction and why it's so fascinating to study this nerdy organ. Then Niklas is going to talk about studies of adipocyte heterogeneity. I'm going to chip in towards the end about some recent unpublished studies. A very, very brief explanation of our future efforts to test our hypothesis. And then finally, just an example of how we try to make ours and others' data publicly available because we think it's important for the scientific community. So let's start with number one here, the introduction to white adipose tissue dysfunction. I think that if you want to remember one figure from this presentation, it's this one. This is the elevator pitch, which basically shows that fat cells are small balloons, and 95% of the volume is constituted by a huge unilocular lipid droplet, which stores energy in the form of triglycerides. It's a very dense matter, but fat cells have a certain capacity to store energy, and when they attain that maximum capacity, they can store much more. So in humans, adult humans, we reach that cap at around 2000 picoliters. So human adipocytes really can't become much larger than that. So what happens? Well, lipids are toxic, and if you can't store them in your adipose tissue, and the best way to put them is in the subcutaneous adipose tissue because that's an enormously plastic organ, an important metabolic sink. Well, if you can't store them in the subcutaneous adipose tissue, they will end up in the liver, giving rise to MAFLD and MASH and eventually hepatocellular cancer. In the pancreas, they will result in both endocrine and exocrine insufficiency, in the muscle, resulting in insulin resistance, in the vessels, promoting the development of atherosclerosis, and finally, if you can't store them in the subcutaneous adipose tissue, lipids will end up in other ectopic depots like visceral adipose tissue. So for us, the visceral depots are really ectopic depots. So think of subcutaneous adipose tissue um, as a safe. So you have to store these very toxic compounds, lipids, in a way where the door to the safe is very easy to open, so you can release the energy when you need it. But it should also be very easy to close. The door and the lock needs to be very well greased. And that's what characterizes a healthy adipose tissue, the capacity to both take up and release fatty acids in a proper manner. A few things about adipocyte formation. Now, remember that contrary to what was believed until 2008, human adipocytes are continuously formed, and they are formed from something called adipose stromal and progenitor cells, ASPCs. These cells, these mesenchymal stem cells, have had many, many names in the past, but now we have a consensus on how to name them, and the consensus is that these prodromal progenitor cells are called ASPCs. Now, sometime during the formation of these adipocytes, they become committed pre-adipocytes. So basically, they can only become one type of cell, and that's fat cells. ASPCs could, in theory, become chondral cells, chondrocytes, or osteocytes, but here, when they're committed, they are committed pre-adipocytes. And this is continuously happening. Approximately 10% of the population of adipocytes is continuously turned over every year. And irrespective of whether you're lean or obese, your adipose tissue can then be in two different ways. So you can be, as you see in the top, with large but few fat cells, which we call hypertrophy, or they can be, as you see down below, many small fat cells. So that's hyperplasia. And having hypertrophic adipose tissue, again, irrespective of whether you live with obesity or not, is linked to risk to develop type 2 diabetes or you have a strong heredity for type 2 diabetes. While being hyperplastic confers a protection against metabolic disease. So the take-home message here is that a healthy expansion of adipose tissue is characterized by um, hyperplasia, functioning adipocytes, low grade, no pronounced inflammation, no pronounced fibrosis, and a normal vascularization. While an unhealthy expansion is categorized by hypertrophy, pronounced inflammation, pronounced fibrosis, reduced vascularization, and of course, poorly functioning fat cells that are characterized by insulin resistance. So with that, I'm going to give the space to Niklas, who is going to talk more about our studies of adipocyte heterogeneity.
All right. Thanks, Michael. Hope that works smoothly and that you all can see my screen. So after this introduction, just want to give you a brief introduction to how we studied the adipose tissue in our lab with the high resolution that we've done before. So we use spatial transcriptomics, the Visium chip from 10x, and this is how it looks like. The top left quadrant there, you have adipose tissue that's H&E stained. And you can place this section of adipose tissue on top of capture probes that are 55 microns in diameter to capture the transcriptome and also histological features of adipose tissue and therefore understand more about biology. So this is what we have done for a few studies that we're going to present. In the first study, we worked intensely with Lovisa from Patrick Ståhl's group at the Royal Institute of Technology. They were one of the co-inventors of this platform, and Jesper Bechdahl is a clinician in our lab who worked on the data together with Lucas Massier from the lab. He has now started his own group, so at Helmholtz also doing similar studies. And so this is what the first data set looked like. We created spatial transcriptomic data on 10 individuals, and this all human white adipose tissue. And what you see here is a UMAP of spots, and each spot is basically a 55 micron spot from the Visium slide. And then what we got out was 20 different clusters. And given that this is spot-based approach, these spots can contain mixtures of different cell types. So in order to understand what the signatures are in these spots, you need to deconvolve the data. And for this, we used single nuclei data from Vijay et al. It's another paper that's published in Nature Metabolism a few years ago. And we could then name these spots so that we saw that they belong to different types of cells. So we had adipocytes, vascular cells, adipocyte progenitor, the so-called ASPCs that Mikael talked about, and then the immune cells. And one thing that we found to be very surprising from this data set was that we found 3 different adipocytes. They're here in different shades of purple: CO3, CO4, and CO6. We've always thought about the adipocytes as a homogeneous, white fat cell with a large lipid droplet, but turns out that we could find 3 different profiles, and we were curious to see what they were. So we plotted their main marker genes, and we tried to name them something so that we don't have to call them CO3, CO4, and CO6. And we named them after their main marker genes. So CO3 was marked by leptin. CO4 was marked by perilipin 1 and 4, and CO6 was marked by SAA1 and 2. One thing to note here is that for those of you who know adipocyte biology, all clusters were marked by adipokines. So that's proteins that are secreted from adipocytes, indicating that they could actually communicate with each other. So adipolep, they secrete leptin, which is a satiety hormone. Adipoplin secrete adiponectin, and the SAAs secrete SAA1 and 2 and RBP4. So we thought this was very interesting and tried to understand more about these adipocytes. And first, we wanted to validate our findings, given that we were a bit surprised that there was some heterogeneity at the transcript level. So we floated mature adipocytes from white adipose tissue by treating the tissue with collagenase and then spinning it. And this floats up small balloon-like cells. So these are the adipocytes. And then we stain them with antibodies, either targeting DDR2 here, which is a plin marker, or it's a adipolep marker, and HSL, which is a plin marker. And then we counterstain with lectin, which stains plasma membranes, and HEX, which stain nuclei. And then we could see that we actually have quite clear heterogeneity also at the protein level. So that warranted further studies of these subtypes and trying to understand what they potentially can do in terms of function in adipose tissue. And the first thing we wanted to check was if they were actually just different sizes of adipocytes, and that could dictate the subtypes. And so, in order to understand if this was the case, we extracted size information from our H&E images. So in the middle here, you have a spot from the Visium slide. It's 55 microns in diameter. And then we cropped images around this spot and then determined size of the adipocytes around this spot. And then we determined the center of the spots and the adipocytes. And then we said that the adipocytes that had the center that was closest to the spot center from the Visium slide was assigned to this spot. So then we could give each spot from the slide an adipocyte size. And this is, of course, an approximation. We can cut the adipocytes at different levels and then get different sizes. But we did this for thousands and thousands of spots. So we think that the average here should anyhow reflect the size. And we compared the adipocytes, the expression of the adipocytes that we could get from the image analysis versus all adipocytes, and we can see that the expression levels of the main marker genes were basically the same. So we're not taking out specific cases of fat cells here in our image analysis. And we could also compare the sizes, the average sizes per donor, either via collagenase treatment, so floating up the fat cells and counting the 100 fat cells in an image versus our image analysis, and we can see that we get exactly the same information. And so from these data, we could see that the adipoplins were significantly smaller than the LEPs and the SAAs, but this differed by I think it was roughly 1 micrometer. So we definitely don't think that this is biologically meaningful. So fat cell size is probably not the determinant of adipocyte heterogeneity. However, we wanted to look at, what's the difference between large and small fat cells, given that we've done this analysis. So we compared for each subtype that we identified, we compared the largest, so the Q4 versus the smallest Q1 of the adipocytes, and then we compared the expressional profiles. And what we could see is that the adipolepts upregulate leptin in large adipocytes compared to small adipocytes. And this is not the case for perlipin, the adipoplins. We didn't have enough observation of adipo-SAA to do this analysis, so we could only compare these two cell types. So just to summarize this first part, we could see that there are 3 different subtypes of adipocytes in the spatial transcriptomics data. We could validate them with stainings at the protein level. We can see that these 3 different identities secrete different proteins. And we believe that given on the size analysis that the LEPs potentially works as watt mass sensors. So when they get larger, they secrete more leptin, and this should then tell the body to not put in more calories, basically. But how about the plins and the SAAs? So in order to understand this more, we actually wanted to look at larger clinical material, but the spatial transcriptomic platform is pretty expensive to work with, and it's difficult to do things in large throughput. So we instead looked at our bulk transcriptomic data. So we reasoned that, we have clinical data I think it's a cohort of 56 individuals, and we have transcriptomic data on these and fat cell information on these individuals. So we reasoned that if these subtype marker genes are clustering with each other, it should be evidence for that, if you have more population of one, then you should have higher levels of all the marker genes, and therefore they should cluster together. And we got that. And then from this, we calculated basically an age and gene score, so that one donor had one number for LEP and one number for PLIN and one number for SAA. And then we could correlate actually the abundance of these different subtypes with different clinical parameters. And one clinical parameter that stood out is the ability of insulin to stimulate lipogenesis in fat cells. So if you have higher abundance of PLIN, according to deconvolution data, the cells are on average more sensitive to insulin. And if you have higher numbers of LEP, it's the opposite. So this gave us an hypothesis that it could be potentially so that the plins are involved in insulin-stimulated processes in human adipose tissue. And in order to test that, we performed hyperinsulinic euglycemic clamps. And I will explain that a bit more in detail. So basically, you take a human research subject. I didn't do this myself, but Mikael did it in the lab together with other clinicians such as Jesper. And then you hook these individuals up to insulin and glucose, and you give them a very high dose of insulin, as you can see in the lower drawing here. And if you do that to an insulin-sensitive individual, the blood glucose levels would drop. So you have to infuse glucose, and then you have to keep them very steadily at a steady state. And for the second hour of this procedure, you can calculate an M value, which is proportional to your glucose infusion rate. So if you infuse a lot of glucose, you have a high M value, and that means that your insulin sensitivity. And the beauty of this method is basically that we could take a biopsy at the start of the procedure. So before we infused insulin, and then after 2 hours of high insulin infusion. And by comparing this, we could understand with spatial transcriptomics, we could understand which cells respond to insulin in human adipose tissue. This is just a volcano comparing hyperinsulinemia over fasting from the procedure. And for those of you who work with insulin, you'll see that there's a lot of known target genes popping up here, indicating that the spatial approach actually worked. But of course, this is looking without the spatial resolution. So we plotted an insulin response score here, going from no response in purple to high response in orange. And here's from one individual where we show the fasting response first. So you can see for LEP, PLIN, and SAA, and also the H and E, that there's a lot of purple spots in the fasting. However, in hyperinsulinemia, the spots remained purple for LEP and SEA, indicating that most cells actually of these cells don't respond to insulin, whereas the plins all become orange. So from this, we propose that actually not all cells respond to insulin. So this changes a bit the view that we have thought about, at least how we have thought about it in the lab, that in a sensitive individual, all your fat cells respond to insulin. So that's dark green here. And a resistant individual, none of the cells respond. And we're saying, according to the spatial data, that there is actually heterogeneity even in sensitive individuals. So only some of the fat cells respond to insulin, whereas others do not respond. However, as a next step, we wanted to understand how this worked in insulin-resistant individuals. In order to do this, we thought that it would be good to have paired samples from an individual being insulin resistant and then becoming insulin sensitive. So in order to do that, we basically did exactly the same procedure as I outlined before. We took insulin resistant individuals, performed a clamp, took a biopsy before and after the clamp, and also did it on the same individuals after they have undergone bariatric surgery. And then this was a 5-year time point afterwards where they become sensitive, again, hooking them up to the clamp, doing the same sampling before and after the clamp. So each individual basically had 4 samples, and the cohort looked something on this like this in average. So over 5 years' time of the bariatric surgery, most individuals lost weight, and then they became more insulin sensitive as shown by the M value. So how about their fat cells? Okay, so we performed both spatial transcriptomics and single nuclei sequencing for this cohort, and this is something that we did together with Nayanika to the left, Jesper and Lucas from the first study, continued on this second study. And here's just a subset of the individuals that we studied. We studied 8 of them, and they reflect the same basically expected outcomes as the large population of the cohort. And here's a UMAP of the single nuke data that we have. We find ASPCs, adipocytes, vascular and immune cells as expected. And if we plot the insulin response score, really the primary response to insulin in adipose tissue is attributed to adipocytes. But even in the adipocytes, you can see that the score is really heterogeneous, going from blue to low scoring to yellow as in high responding cells. So this, sorry, this mimics the spatial data that we have. We just use another method to see it. And if we plot every nuclei that belongs to an adipocyte before versus after the surgery, again, we can see that there is really a high heterogeneity in the response to insulin, but that weight loss certainly improves the response. And if we want to look at individual donors, it looks like something like this. And sorry for this, this is really a messy graph, but the point is that there is really high or pronounced inter-individual differences in the effects of weight loss in terms of the insulin response. So you can look at subject 4, and you can see that subject 4 basically improves a lot when it comes to weight loss. But if you compare it with subject 7, basically they look exactly the same at time point 0, but subject 7 does not improve at all. So there is a really pronounced inter-individual differences in the effects of weight loss. So from this second part, we just want to highlight that we thought that the adipolapse or wet mass sensors, and we think that we have pretty ample evidence that the adipoplins are some type of insulin responding cells in the tissue. Mikael, you want to take over?
Sure. We're interested in understanding whether there were also differences in the proportion of different subtypes of cells of adipocytes across different depots. And to this end, we collaborated with Matthias Blyer in Leipzig and his group, and we obtained subcutaneous adipose tissue, but also tissue pieces from the same individuals, but across different depots in the gastrointestinal tract, going from omental adipose tissue, which is this fat curtain we have in front of the intestines, small intestine mesenteric adipose tissue. We went to large intestine mesocolic adipose tissue, and then finally epiploic adipose tissue, EPI. You see the acronyms that we've used. One thing that one should mention here is that these visual depots are located across different parts of the gastrointestinal tract, where epiploic fat is the depot that is closest to the microbiome, because epiploic fat is basically these big fat pearls that are prominent on the colon. So they're very, very close to the microbiome. And this is different from, for instance, omental or small intestine mesenteric adipose tissue. So we obtained these samples, and this is work done by Jutta Jalkanen, who just defended her thesis, and Jawe, who was also a very gifted PhD student like Jutta, with different expertise. These are the 8 subjects, 4 men and 4 women. They had different BMIs. They were all living with severe obesity because they were undergoing bariatric surgery. They were all middle-aged. They had different HbA1c and very different insulin sensitivities, as you can see. Now, the analysis pipeline was the same as we did before, that Niklas went through. So it was single nuke and these spatial transcriptomics Visium slides, and we did hefty bioinformatic analysis. If you look at the enrichments of specific cell types at the class level, you may appreciate that ASPCs constitute approximately 20 to 30% of the proportion of cells in all the 5 different depots. Mesothelial cells are only present in the visual adipose tissue, so that was a big relief that we didn't see them in subcutaneous adipose tissue, because in that case there would have been something strange. Vascular cells constitute somewhere around 7 to 15% of the cells. Immune cells in these people living with severe obesity, approximately 30 to 40%. And adipocytes, as you know, are only constituting approximately 20% of the cell population. We also saw some Schwann cells, but please note the y-axis there, that it's very, very low, the proportion. Now, one thing that we noticed was that if you look at individual cell types within each class, you can appreciate that subcutaneous adipose tissue has 3 enriched ASPCs and several vascular cells that are really enriched in the vascular cell. Several vascular cells that are enriched in the subcutaneous depot. Now, if you look to the right, you see these 20 of 28, these are immune cells. And the blue are myeloid cells and the purple are lymphoid cells. And you can appreciate one thing that we noticed here is that there's a high proportion of myeloid and lymphoid immune cells that are really enriched specifically in the epiploic depot. So this made us interested because then we figured that maybe there is something about the epiploic fat that really makes it stand out from all the others. One big advantage of using spatial is that you can also understand more about other cell types. Here is just one example where we have the H&E stain and where we can see waves of differentiation, adipocyte differentiation in the tissue. Here are some early pre-adipocytes, ASPCs. They're found in these stromal parts of the tissue, the reticular tissue here, and then they become, as they progress in their differentiation, you see that they go out in the tissue, and the committed pre-adipocytes are much closer to the adipocytes, the mature adipocytes. So this underscores the fact that adipose tissue is much more organized than what was previously thought of. Another thing we looked at was, of course, the different adipocytes. So we found, and we were happy to see that we found the LEPs, the PLINs, and the SAAs that we had described before. And if we looked at the marker genes, the top 3 marker genes in this Jalkanen paper versus the Bechdahl paper, that was the published paper that Niklas showed, we could see that all the major marker genes were congruent across the studies. Now, one thing I want to emphasize here are the adipo-SAA, because as you remember, the adipo-SAAs were the only cells where we didn't really understand their function. And you can see that SAAs are marked by SA1, 2, and SES1 genes. And if we looked at the proportion of these, the enrichment of these genes in the different depots, you can appreciate here that the the enrichment is really much higher in the epiploic fat, and that's true for spatial transcriptomics. It's true for single nuke RNA sequencing, and also at the proteome level, although I don't show that here. So the epiploic fat seems to be enriched not only for lymphoid and myeloid cells, but also for SAA adipocytes. So we didn't perhaps say so much about this previously, but you can also appreciate that the SAAs tend to cluster in the tissue in these homotypic clusters. So they tend to like each other and stay together as buddies, while the LEPs and the plins are more, interspersed across the tissue. We hypothesized that the fact that there were a lot of SAAs and a lot of lymphoid and myeloid cells could suggest that there are a lot of interactions and communications between these cell types across the different depots, in particular in the epiploic depot. So for this, we used CellChat, which is a bioinformatic pipeline that predicts communications, intercellular communications in a tissue. And you can see here that the number and the strengths of these intercellular communications were much, much stronger, and they were many more in the epiploic fat compared to all other depots. And this is a busy slide, but it basically shows on the x-axis what tend to be senders, and on the y-axis what tend to be receivers. And just start up in the left corner, you see the ASPCs. They tend to be across all depots along the regression line of 1, meaning that ASPCs in these different depots tend to be both senders and receivers. This should be compared with, for instance, the vascular cells up to the right, where you see that they mainly tend to be receivers of signals rather than senders because they're more to the left. Now look at the adipocytes down to the right. You can appreciate that they seem to be senders, particularly in the epiploic depot. And if you compare that with the myeloid cells, they tend to be receivers, and particularly in the epiploic fat. So we hypothesized that there might be communications going from adipocytes to myeloid cells in particular. And for this, we depicted these charts here where you can see the strengths of the different intercellular communications, and the color indicate that there is a strong signal going from the red adipocytes. And in the epiploic fat, these fat bands of red lines go particularly to the myeloid cells, suggesting that there is in fact a communication between adipocytes and myeloid cells. So just briefly, this is nothing to do with our paper, but there are papers in the literature suggesting that the microbiome releases factors, for instance, LPS, endotoxin, that affect the production of SAA, and the SAA binds retinol, and retinol is very important to form retinoic acid, which in turn is very important for adaptive immunity as it activates a number of different immune cells. So we hypothesized that microbiome-derived factors could affect neighboring adipose tissue, so epiploic fat, and thereby promoting SAA expression, which in turn modulates immune cell activation. So I'm going to just show you some very top-line data here. So we tested in adipocytes, ASPCs, endothelial cells, and THP1 macrophages whether LPS could induce SA1 expression. And we found that it actually did, but only in the adipocytes. So only adipocytes responded to LPS by promoting an increase in SA1. And if you used SA1 and treated the stromal vascular fraction of adipose tissue from human donors, only the myeloid cells responded to SA1. So to complement this figure, what we suggest here is that LPS released from the microbiome in the colon results in an increased expression of SA1, specifically in adipocytes in the epiploic adipose tissue, which in turn seems to be important for immune cell modulation of myeloid and lymphoid cells in epiploic adipose tissue. So to complement the figure that we showed previously with the watt mass sensors and the watt insulin responders, we could then suggest that SAAs are adipocyte or adipose tissue immune modulators. And we're testing these efforts in different interventional studies where we, for instance, test the effect of oral antidiabetics, but we're also comparing the effects in different populations with or without cardiovascular disease and type 2 diabetes. And finally, my last slide before acknowledgement here is that we are both very committed to make our data available to the scientific community. And for this, we have developed the Adipose Tissue Knowledge Portal, which summarizes almost everything we have generated over the last couple of decades. So here you can put in your favorite gene or genes and obtain summarized information in a very easy format that you can use for your publications. So it's already in a publication-ready format. The only thing we ask you is to acknowledge and to refer to the paper, the original paper where this is published. We have included 67 data sets from more than 6,000 men and women, and we've also put in basically all our in vitro data that we've ever generated, and you can compare expression of genes in different depots. You can look at single cell expression. You can also look at the spatial transcriptomics and also define whether specific genes are enriched in specific cell types in your in adipose tissue. Also, if you want to define, we have a gene finder module. So if you're interested in finding genes that meet specific criteria, for instance, expressed in women in the visceral adipose tissue of women with high HOMA-IR, high insulin resistance, then you can put these cutoffs in and get a list of genes that fulfill these criteria. Now, when we published this, it turned out that Evan D. Rosen's group was developing a similar effort, but from a different angle. And they've developed the mammalian adipose tissue knowledge portal, which focuses on genes in different species and in many, many more depots than we had. So we are now developing a common entry portal so that you can access both adipose tissue portals, but they are complementary. So we're not competitors in this area. We're actually complementing each other with these two portals. So finally, this is a large collaborative effort. So apart from Niklas and all the people that we've mentioned throughout, we didn't have time to mention many of the other people that have generated data and published really interesting results over the year, and all our collaborators and funding bodies. We also want to thank all the patients and research participants, of course, that have contributed so much over the years. Thank you very much.
Thank you very much for this really, really nice presentation and for sharing all your data with the community, which is a really great opportunity for everyone to focus on something that might be really difficult to get without all the funding, and it is really great. So it is now opened for discussion. I believe we have the first question here. LPS has been suggested to cause endothelial dysfunction in various cell types. Did you also notice an effect on endothelial dysfunction in these adipocytes causing higher accumulation of lipids?
So I think that the simple answer is no. We have not studied this. I think the entire LPS SAA axis is extremely interesting. As you may know, SAAs are also closely linked to cardiovascular disease and atherosclerosis development over the years. So we think we're onto something, although we're not sure how exactly this works. We do not have the expertise to study endothelial dysfunction. We need to do that in collaboration with other groups. We did not see, as you saw, an increase in SA1 when we treated them with LPS, that doesn't mean that LPS doesn't have effects on endothelial cells. Quite the contrary, it certainly has, but we have not studied that. So that's something we hope to be able to do in collaboration with experts. And we're working, for instance, with Joanna Kaluka in Aarhus, trying to understand the role of the vasculature in adipose tissue. But I think it goes for both me and Niklas that we're real fat cell nerds. So that for us, everything revolves around the fat cell, and you can only so much.
Okay, great. Thank you. So we have another question from Anna Gianconero, who is saying that very interesting talk. And have you investigate other microbial metabolites?
The answer is no, we have not.
Okay.
And I think that that would be a whole research field of its own. So what we're trying to understand now, many may know, the role of creepy fat, which you see in inflammatory bowel disease, where the fat is um, located, there's an inflamed fat close to the inflammatory regions in inflammatory bowel disease. And so clearly, adipose tissue must have a role in protecting us against infections and communicating in some way with the microbiome. By the way, epiploic fat does not exist in rodents. So this is something that is specific for us humans and primates, which makes it even more interesting. And we're now trying to understand how this works, but in collaboration with researchers in Manchester who can actually get access to adipose tissue from individuals with inflammatory bowel disease undergoing surgery. And we can take them from the affected and the non-affected area to understand whether there are differences in these SAAs, for instance, and how they react to different inflammatory cues. And it's very interesting I mean, as we know, how much has unveiled on the role of the microbiome. And of course, there is a big debate on how important this is, and there are believers and non-believers. And we're somewhere in the middle, I guess, at the moment.
Okay. There is also another question, this time from Elisa Villalobos. Wonderful work. PLIN1, AdipoQ, and LEP are classic genes to annotate adipocytes in omics from other tissues. However, your subsets were differential expression of those. What would you recommend when identifying adipocytes in omics from extra adipose tissues?
So maybe you want to take that, Niklas, or?
It doesn't matter.
Yeah. So all of these are wonderful markers for adipocytes for sure. Plin in particular, in particular is very good. Um, Remember that we're now talking about enrichments. This does not mean that there is no lipe or adiponectin in the LEPs. It's just that they're enriched in these cell types. And we have observed this using spatial transcriptomics, but this is not unheard of. Silvia Corvera has shown similar differences in a completely different model system where she develops different adipocytes in culture from murine and human samples. And she can also get these fat cells that tend to be enriched in leptin versus adiponectin. So we think that there is a heterogeneity, but it's not 0 or 100.
So the short answer is that you can continue using plin, adipose-Q, and leptin. And if you want to talk more about subtypes and understanding if there are different proportions of subtypes when you use it in extra adipose tissues, you can always use the marker gene panels that we show in our papers. For instance, the Bechtel paper.
Hey, excellent. And okay, then we have another question. So a young Yu asks, how could diet microbiome interaction play a role in driving the formation of the white adipose tissue? And how can these findings translate into weight controls?
Yeah, so as you know, there is a debate about the role of the microbiome in weight control. So Max Newdorp is, to my knowledge, still the one that leads this field. We're collaborating with him and we're discussing a clinical trial on this. He has done the fecal transplantations in man, so in humans. The effects of fecal transplantations as a giving healthy microbiome to people with obesity and insulin resistance. These effects are very transient, and they're quite small if you compare to the drugs that we can use in daily practice. So I think that um, there are individuals that um, may benefit more from this, and these are usually the ones that have a microbiome with a low diversity. So people that have a very diverse microbiome, they tend to respond less to fecal transplantation. So it's more about, and we know that low diversity of microbiome is often a sign, is often linked to metabolic disease. So long story short, I'm a little bit unsure with how the diet and the microbiome interacts. I think it can be of importance for some individuals, but I don't think it's important for everyone. Remember that humans are among the creatures on Earth that are the most diverse in what we can eat and survive on. I think only rats are more or less, in the same ballpark when it comes to food diversity and the fact that we can be perfectly healthy using very, very different diets, suggesting that we are very prone to adapt to specific diets.
Okay, excellent. I would actually have one question, and that is like, do you think like this different adipocyte phenotypes, so can interventions like GLP-1 agonist or current obesity drug treatments like modulate the or do you have like plans in doing a research on this?
Yes, that's exactly what we're doing right now. So that's an obvious question. So that's exactly what we want to know. We're particularly interested in uh, third generation, second and third generation drugs, multicretins in particular, whether they have specific effects. So yes, in short, we're doing that, but we don't have any results yet.
And then another question also that do you think there will be other like subpopulations or how many could there be? Because this is validated, you have 10 individuals and then validated data now for these subtypes, but will there be more? Or what do you think?
There are so many subtypes discovered in every single paper, and if you look at the single nuclei RNA sequencing data sets that are out there, it's over 20 of them now that are based on human white adipose tissue. And everyone proposes 7 or 8 or whatever subtypes, but they are basically never overlapping. So there's zero consistency in the heterogeneity found among adipocyte identities comparing across studies. And the lack of, consistent heterogeneity, I find to be very problematic for the field. We have undertaken pretty ambitious approaches with like meta-analysis comparing across studies, and we really, really don't see overlaps in the marker genes from different studies. However, when we do the spatial transcriptomics, it doesn't matter if we do it here in Sweden or if we do it on samples that are obtained in Germany by completely different surgeons and the data is generated there. We always get these three fat cell identities from the spatial data, which brings us a bit more confidence. But still, I think really we all need to work on this together, all research in adipose tissue to try to understand this better.
I also think it's important that we should, because this tends to become very much transcriptomic mumbo jumbo, and we have to link these subtypes to functions. And that's what we try to do in our efforts in situ, in humans, trying to understand what these fat cells actually do. Because that's the big issue right now. When it comes to fat cell studies, it's still 2025. We can send people to the moon, but we can still not efficiently isolate mature fat cells at the single cell level and functionally study them. There is no way of fact sorting mature adipocytes that is actually working. Um, a few efforts have been made. Kirsty Spalding, for instance, she had a postdoc who mouth pipetted 1,034 or whatever adipocytes. It took her, I don't know how many hours. I don't think we can ask people to do that. That's something you do once in your life and never again. So that's the big conundrum right now, that we cannot study fat cells at the single cell level functionally.
Okay, thank you. I have a question as well. It's about adipose tissue health. Do you think that one day it can become a measurable clinical parameter, like similar to hepatic or renal function, and it might be useful to inform cardiovascular disease prevention?
The short answer is yes, but it cannot be based on biopsies. Fat biopsies will never be part of clinical routine. Me as a clinician, the only thing we can have are simple measures like blood tests. So based on these data, we can find biomarkers that are circulating and that are linked to different phenotypes. And that's exactly the type of efforts we're doing now with longitudinal and cross-sectional studies, trying to link fat cells and thereby markers and biomarkers in the circulation to different clinical phenotypes. But I mean, working with adipose tissue, and Sini knows this as well, is I mean, there's a small subset of very nerdy people in the world that actually tend to do that. The rest go for muscle.
Yes, it has its own challenges, but then also it can give a lot, right? So
Yes. I mean, we love it. So obviously both me and Niklas got stuck to it. So there is something to it that is fascinating. The fact that it's not easy to study, that's also one of the big challenges, and that's why we want to contribute to this field.
yes, excellent. Then we have another question from Elisa Villalobos, actually. If you look lipidomics in plasma, what fat depot would be the one with more similarities?
So we are doing these lipidomics analysis now. We're doing them at Steno because they have a very nice facility there. And we're doing both adipose and plasma lipidomics. I would guess that, on average, it would mostly reflect subcutaneous because that's the biggest, just by mass, it's the biggest depot in most Caucasians living with obesity. That doesn't necessarily translate to the same thing across all ethnicities because we Caucasians tend to have a capacity to increase our subcutaneous adipose mass very much compared to many other populations in the world. So I think the jury's still out there on what the best lipids would be. I hope we can get back and answer that question in a couple of years when we have all the data. We've just sent the first batches of samples down to Denmark, so that's also one of the great things of this research field. There are so many questions that are unanswered compared to many other fields.
Yes, excellent. Uh, Then I would maybe still ask like, what about the microenvironment? Like you have shown data from different depots and then the spatial data, but much do you think that the adipocytes are affected, but how vascular is the tissue or the endothelial cell interactions or immune cell interactions and maybe you have also plans to study this further.
We think that there are many more spatial features of adipose biology than what people originally thought. So we find that the adipocytes are dispersed in different ways, like Mikael showed on one of his slides. But we can also identify, for instance, organization of the adipogenic trajectory in layers stemming from CD55 progenitor cells in the reticular interstitium and then growing out into the tissue. So there are many of these aspects that we're trying to look into even further, and we believe that we need to follow up on some of these unbiased approaches that we've done with supervised machine learning to really see clear-cut patterns in terms of spatial aspects.
Okay, yes, thank you so much. So I suppose we
No additional questions.
no other questions at at this time, and I think we are nearing the end of our 1 hour here. So we want to thank you really much for this excellent talk and really interesting topic and your work on it. And we were really happy to have this webinar and also to share it to all the audience in Early Career Academy and Diabetes Association.
Thanks for inviting us.
Too much
Thank you.
and
Good luck.
see you soon.
Bye-bye.
Thank you
Bye.
very much. Bye.